So the next talk, I'd like to introduce Allie Carson, and you all know Allie from the BC Cancer Agency, and Allie's going to be speaking today on his project, the genomics of applied to the management of high-risk AML and myelodysplastic syndrome. So again, this is one of our PMP projects, and that has just initiated recently. So Allie, thank you very much.
I'm speaking on behalf of a team. We're listed here, so we have myself, the director of the Cancer Genetics Laboratory that does all the molecular genetic testing for cancer in the province of BC. Mark O'Mara, who's the director of the Genome Sciences Center. Peter Chow-White and Andrew Feenberg from SFU, who are involved in the social sciences and humanities part of the project. Donna Hogue, who is a leukemia BMT hematologist at the Vancouver General Hospital and the BCCA, and she does AML clinical trials. That's acute myeloid leukemia that I'll be talking to you a little bit about today. And then Steve Jones is heading the bioinformatics, and Stuart Peacock, the health economics. So the problem is shown, I think, quite acutely on this slide. The number of cancer genetic testing are increasing. There's been a threefold increase over 5 years, but the budget has been relatively static. So we've been thinking about this problem, and I took on this position that I have now about 2 years ago. And basically, we've been held back from moving forward because there has been no budget. And so we are looking for solutions. And one possible solution is that the cost of sequencing is declining using new generation technologies. And we do a lot of diagnostic, not all the diagnostics, but a lot of the diagnostics is based on sequencing. So here's a potential solution. Take on the new equipment, the new technologies to deliver testing. And the Provincial Health Services Authority recognized the issue. They funded us to develop a high-throughput sequencing center, which is called the Center for Clinical Diagnostic Genomics, and the mandate is to provide sustainable and scalable clinical diagnostic genomic testing for the population of BC. So this is PHSA funded, but we've received a lot of support from Genome British Columbia to really bring this into the clinic, as well as the BC Cancer Foundation. The center is a partnership really between the Genome Sciences Center and the Department of Pathology at the BC Cancer Agency, and it's housed in the Genome Sciences Center. So the reason that the cost of sequencing has gone down is really that the capacity to sequence has increased. So the ability to sequence a lot more, very quickly has increased, so that the cost for a single letter of the DNA code to get that information is much cheaper. So that decrease in cost really doesn't add value unless you can use the capacity. And there are a couple of ways you could use the capacity. You can sequence specific things. Dr. Levin mentioned it's a no-brainer to sequence BRCA1 and 2, which we're currently doing using the old technology of Sanger sequencing. But if you were able to use the capacity and pool a number of patients into a single instrument, you can then gain cost efficiencies. And in fact, we have a project that's Genome BC funded that we will be delivering this kind of testing in the spring. The other project, which I'm going to spend more time on today, is that of looking to see whether we can look at a lot of genes now in one or few patients, rather than looking at a few genes in a lot of patients. So the current projects out of the Center for Diagnostic Genomics include BRCA1/2 sequencing, as I described, and we've spent almost 2 years validating the process, the instrumentation, and we will be delivering this testing now on the next generation equipment in the spring. So we're just completing the documentation. The cost per test has been reduced, and the capacity is greatly increased. So we can serve potentially more than this province to do the testing. But even if we're only to serve this province, we're still doing it cheaper than we will be doing it cheaper than we currently are. As part of an expansion of this project, we are also looking to sequence a panel. These are highly penetrant genes that predispose to cancer. We've developed a panel in collaboration with medical geneticists as well as the genetic counselors at the BC Cancer Agency. And this is another Genome BC funded to deliver this type of testing in a single platform. What I want to talk to you about today is to look across now the genome, to look at all the DNA and or all the RNA to use as a diagnostic in acute myeloid leukemia. And so we will be doing, we're currently engaged in a retrospective clinical trial of bank specimens. The project is integrated with addressing ethical and health economic issues, and we expect to begin a prospective trial this summer with the delivery of the test in the next year. Because the mandate really is to provide this service for various types of diseases and various other labs, we're happy to work with anybody who's interested in using this capacity now to deliver genomic testing. So the disease I'm talking about today, acute myeloid leukemia, it doesn't affect a lot of British Columbians, but this project really is going to deliver a proof of concept. We hope that this kind of approach is feasible and cost effective. The treatment options currently for a patient with AML is either chemotherapy, intensive chemotherapy, or stem cell transplantation. But without treatment, patients will die within a matter of weeks, so it's a highly aggressive disease. While stem cell transplantation can be quite successful, it's very expensive, in the order of $200,000 per patient, plus or minus $50,000. It's high risk and sometimes fatal, so the toxicity of the treatment is quite high. However, by stratifying patients to chemotherapy or stem cell transplant, you can improve the outcomes in a major way, and I'll show you some of the guidelines that have been developed already by multiple international groups. The stratification is based on genetic markers, and the problem is that the number of markers is rapidly increasing. And so the goal of this project is to improve patient outcomes and reduce overall costs to the health system by improved testing. And by overall costs, I don't just mean testing costs, but also the improved outcomes, which hopefully will have not just human value, but also physical improvements. So the major goal, the primary goal now is to stratify patients with AML to the best treatment option, stem cell therapy or intensive chemotherapy. There are secondary outcomes. We hope during the process of this project and ongoing to be able to now identify pathways that are operative in patients who are maybe not candidates for either one of those therapies, so that we can repurpose existing drugs. And then finally, I think a major achievement would be to provide proof of concept for genetic testing in other cancers. So these are the National Comprehensive Cancer Network guidelines of how to stratify patients. And there are three risk groups, a good risk group, an intermediate risk group, and a poor risk group. The good risk group would receive chemotherapy only. The poor risk group receives stem cell transplant, but there's a large proportion of patients who fit in this intermediate risk group in whom we don't know whether to transplant or treat with chemotherapy. However, if you use cytogenetic testing, you can now stratify better these patients who are going to be low or high risk. And then if you, in addition, use if you use testing for 4 additional genetic abnormalities, you can stratify this intermediate risk group into either a good risk group or a poor risk group. And so here's some of the data that locally that's been reproduced worldwide, really, that if you have this mutation, an NPM1 mutation, and do not have a FLT3 mutation, you actually do quite well. Whereas if you have a FLT3 mutation without an NPM1 mutation, you do quite poorly. And what's also been shown, and I'm not going to show you the data, but it's well described, is that if these patients here are transplanted, then their outcome is much better. So this is showing probability of survival over time. Now, as I said, the number of markers is rapidly increasing. So what you see in yellow is sort of karyotyping, which we do here in BC. We also do 2 of the 4 markers that are suggested by guidelines to be doing in BC. And here's a whole host of new types of tests that are under evaluation or have been recently described as being predictive in this disease. And so this project is really aimed at trying to preempt this onslaught of testing that we're maybe asked to be doing in the coming years. And in fact, to catch up with current guidelines as well. And so there are 3 aims in this project. One is to develop and understand 2 genomic approaches, one using DNA, the other using RNA, to identify all the variants that are present in AML, which will then allow the leukemia BMT physicians to choose the best therapy. We have, as I said, a health economics team led by Stuart Peacock, who will be examining the economic implications of genome-wide testing, and as well we will be looking at developing a strategy to guide regulators, which is very important, as well as clinical and laboratory physicians on how to manage this information ethically while still respecting privacy law. So, in order to start this, we needed some feasibility data. This project is a lot less mature than what you heard from Bruce and Paul earlier. So what we needed, we had clinical guidelines that there were a number of genetic tests that needed to be done, but we didn't have data that said it's feasible. And so what we did is we looked at the Cancer Genome Atlas Project, which is a consortium of many genome sequencing centers across the U.S. The Genome Sciences Center is the only international site in this project. And what we're looking at the Genome Sciences Center is the mRNA and the microRNAs for a variety of cancers. And in particular, a number of acute myeloid leukemia samples had already been sequenced. So we looked at this data. This is a slide from ND Mongol at the GSC, to see if we could detect some of the karyotypic, the cytogenetic abnormalities that are present in AML that guide therapy. So these are translocations of one part of one chromosome onto another. And so by sequencing, you can see where the breakpoints of those chromosomes occur to determine whether there's a fusion. And so what would be described in classical terms as a translocation of one chromosome to another can be described as a gene fusion using sequencing. And what this slide shows is that we can pick up all the known, in this sample of 179 patients, known AML fusions, but we can also detect fusions that have not been described anywhere in the literature. We can also find point mutations or small insertions or deletions in the sequence using this cohort of patients. And when we compare known variants, known mutations that are present in this cohort from the TCGA, the proportion is similar to what we see in our own cohort of patients here in BC. So these are different data sets, but the proportion suggests, yes, this is a feasible approach, that we will be able to pick up what we need to. In addition, we can pick up variants here, mutations that we're not currently testing for, but should be. And these are sort of markers in the pipeline that other groups have described, and some that have been described here, that suggest some of which have a predictive value.